Okay, the next presentation will be done by Yaroslav Pirko from the Institute of Food Biotechnologies and Genomics. Good afternoon, dear colleagues. Allow me to present our work, which is development of research and industrial technology synthesis of dot nanoparticles using phytochemical tanks and plant cell culture for visualization of subcellular structure. The supervisor of this project was director of the Institute, Yaroslav Blum. It should be noted that our project should be divided in two parts. First is nanoparticles of metal, silver, and gold synthesized using the phytochemical system. And second part is synthesis of cadmium and zinc sulfide quantum dot nanoparticles with biological systems, phytochemical tanks, cells of plants, and fungi. What is the product that can be created? Synthesized nanoparticles have the same criteria as the most of the nanomaterials. Nanoparticles of metals, silver and gold, and quantum dots are made with the help of various biological systems, in particular phytochemical tanks that plant extracts, tissues, and cultures. With the help of cultural liquids, culture fluid of bacteria and fungi. How does this technology improve the products, the existing and known products, and create a new class of products? The biosafety of synthesis of nanoparticles according to the developed technology as opposed by chemical synthesis using the more toxic substances. We have samples. These are silver nanoparticles obtained with phytoextracts of various plants. TEM microscopy show that they have a size of 5 to 25 nanometers, have a spherical form. And this slide shows nanoparticles of gold obtained with a similar technology with phytoextracts. And the size of our nanoparticles is close to 25 nanometers. This slide shows cadmium sulfide quantum dot nanoparticles obtained with the help of this kind of vessel, this tank, with use the fungi, Pleurotus substriatus, which is very widespread fungus used in food industry. This technology itself. Synthesized nanoparticles have some certain chemical and biological properties. The shape and size of nanoparticles are determined by the properties of biological systems, namely substances that are included in them. Depending on the taxonomic status of biological objects, the conditions of their treatment, the concentration of the components, we can obtain the nanoparticles with specific physical and chemical properties. And also, the substances in the biological systems are both reagents involved in the synthesis of nanoparticles, and at the same time, they are substances that prevent the destruction of the synthesized nanoparticles, so they conserve them. How these particles can be applied? Currently, we have tested our nanoparticles, the quantum dot nanoparticles, for fluorescent microscopy, as you can see on these slides, improved visualization of some subcellular elements. And for instance, nanoparticles of silver and gold can be used as antimicrobial agents.
Інтелектуальна власність. На сьогоднішній день ми маємо одну заявку на корисну модель.
And this is the application for patent that we filed, process for the preparation of nanoparticles of silver and gold. The invention relates to the field of nanobiotechnology, where nanomaterials can be, based on silver and gold, can be used as a hygienic and antiseptic in medicine, as well as for the future manufacturing nanodevices for medical science and biomolecular studies. Now, what in particular is going on with this project? We obtain nanoparticles that meet exact criteria, size of nanoparticles up to 100 nanometers, stability. And another important trait is that these nanoparticles have a very long life cycle. And this technology allows to obtain nanoparticles by a more eco-friendly process, which matches the green synthesis process, unlike the more toxic and energy inefficient processes. And using the new methods of obtaining nanoparticles, which is phytochemical tanks and culture fluid of bacteria and fungi. Now as for the market, where can it be used? Nanoparticles of gold and silver had been used for many years as antiseptics, so this is the potential market. Also, these particles can be used by institutions part of National Academy of Sciences, the Ministry of Health, some pharmaceutical companies, and we also have an example of a cooperation with company ECMA, which deals in making clothing elements and accessories for severe restrictions as to their clean environment, and the shoe industry, or special shoes. So 1.5 million per year is the estimated volume. Also, we're looking forward to having some bank loan for further perfection and development of the process of synthesis and further studies of antimicrobial properties of the gold and silver nanoparticles and implementation of the results. So thank you. Now, as far as I understand, so you have biopolymers after the extraction, which support the growth of nanoparticles of gold. Is that right or so? Yeah, they not only support, they actively are engaged in this process. So it's a regular chemical reaction, and they stabilize it. Yeah, I understand. I understand the process, but the main problem with phytoextraction is the following. When you do a phytoextract, it's very hard to make it, basically, because you either have to use ethyls or thermal phytoextraction, or whichever way do you use. How do you coagulate biopolymers? We cannot say currently what exactly. Is it biopolymers or smaller moleculars that are extracted, polysaccharides? And we do extraction with water. We don't use ethyls or other chemicals. But the output is small. Yes, it's small, but it's sufficient. And we were surprised. So the advantage of this technology is its simplicity. Okay, then we will privately talk about it further. Okay, question from the back of the room. What are the components used in the culture fluid to restore the compound?
Ну, значить,
To lead it to metal state. The culture fluid, okay, if we use silver, we don't add anything because phyto extract contains nanoparticles of silver. But whereas fungi, we use to obtain quantum dot particles, cadmium S. If we took Fusarium fungus, it has sulfate reductase signal. So in this ferment system, it degrades cadmium SO4 to cadmium S. So where do you get their ions of silver and gold in your system? We artificially add them. How? As salts. Soluble salts. Okay, but salts will negatively impact the bacterial cells if you add the bacterial culture fluid. But first we grow the bacteria, then we collect the culture fluid, and then we add. So you take the cultural fluid or culture environment. Fluid is the environment where bacteria had grown. Yeah, right. It's the know-how. Maybe I did not speak in a specific term, but nanoparticles that we synthesize with the help of natrium citride. We synthesize the several nanoparticles, and they are stored in the regular conditions. Those nanoparticles that we synthesize with phytoextracts, there are examples where for a year we have stored a sample. And of course, if you mix it again, the same result will ensue. The microphone was not on. We are studying this problem, the synergy effect. The phytoextract of the particles that are in a phytoextract, and the nanoparticles of silver. So when you obtain them, your nanoparticle is surrounded by. Вмикайте мікрофон, будь ласка. Виступаючи.
Але, подивіться, ми зараз проводимо дослідження стосовно того.
So we're doing a research as to there's an antimicrobial action nanoparticle, and when we have these nanoparticles obtained with phytoextracts, we have to say that not the remainders of the phytoextracts impact that, which have antimicrobial properties, but nanoparticles that were synthesized during our studies. Okay, if you take silver sulfate or nitrate, what happens with the anion? It is washed away. Okay, thank you. We thank you for the answers and the questions.